Hello there, ladies and gentlemen, and welcome to Unit 4, the structure and function of cells. Here in our first video lesson, we're going to be analyzing three major topics dealing with cells. I guess you can call this video an introduction to cells or cells at a glance. Okay? We're going to start off by examining the history of the cell. All right? A little bit about discovery, a little bit about microscopy. They kind of go hand in hand. All right, so that's the first video topic, a little history and discovery of, a little microscopy of the cell. The second major topic we're going to get into is the cell theory. So what looking at cells and discovering cells and gathering the information of or about cells early on led to. Okay, so what some of this early discovery, interest, and investigation led to, the cell theory. That's our second major topic in the video today. And last but not least, the third major topic is going to be two common types of cells. Eukaryotes versus prokaryotes. Okay, so we're going to talk about the two major types of cells, common features that each one of them share, and then what's the major difference? Why do we have two types? All right? Without further ado, though, let's get right into this thing. Way back in Unit 1, we talked about some of the unifying themes of biology and some of the traits of life. All right, that set of criteria that an entity needed, okay, to end up being deemed as living. So you had to possess, or at one time possess, all 7 of those characteristics in order to be deemed a living organism. When we defined cells in that unit, we stated that they are highly organized, tiny structures with thin coverings called membranes, and that they are the smallest unit capable of life functions. We're keeping that same definition as we're going forward here in Unit 4, but I like to throw a few more items at you. To me, I define a cell as the smallest unit of structure and function for life. Okay? It's the smallest structural and functional unit of life. You'll hear me refer to it like that over and over and over in this unit. I also like to use the phrase self-sustaining. Okay? So with a cell being the smallest structural and functional unit of life, it's also self-sustaining. Okay? It's the smallest unit capable of carrying out daily life function and sustaining itself. That's what a cell is. All right? Now, we didn't know about cells for our entire existence here as a human race. Okay? We started to get into these items and discover these things way back in about the 1600s. All right? That's when we first started to open up Pandora's box in regards to cells, the cellular level of life, and also the molecular level of life. Robert Hooke was the first individual with a very crude microscope to observe these itty-bitty tiny little boxes that he saw in a thin slice of cork. They resembled what looked like little cells at which monks would live in a monastery, itty-bitty tiny rooms. So he called these little small structural blocks and boxes cells, hence how the term cells was coined. He was looking at a remnants of the smallest structural unit of life, the actual cell there. Okay, he was looking at dead plant cells of that cork. A few years later, Dutch scientist by the name of Anton van Leeuwenhoek, with a much more refined and that's what I'm looking for here, capable microscope, actually observed some of the first microscopic animals. He called them animalcules. He basically put a little droplet of water on the end of this tiny little microscope. This piece of metal about the size of a credit card had a little itty-bitty spherical lens inside of it, and it had this little pointer right in front. Okay? He put the drop of water on there, he observed through the spherical lens, and he saw these itty-bitty little microorganisms moving around the water. So it looked like stuff that you would see walking down the streets or out in the fields, okay, or flying around through the air, except it was in that tiny itty-bitty drop of water. All right, and we're talking the 1600s here. All right, like 1675, 1677. This guy was seeing these tiny itty-bitty little things move around this drop of water. He called them animalcules. That was Anton van Leeuwenhoek. Okay, Van Leeuwenhoek is kind of referred to as the father of microscopy. All right, the the section of science that was all about utilizing lens technology to magnify that microscopic world. All right? So early on, the discovery of cells, okay, or the coining of the name cells by Robert Hooke, and the discovery of these animalcules, these tiny bitty microorganisms, by Anton von Leeuwenhoek. All right, early on cellular discovery and investigation by these gentlemen and other scientists in the field at that time, okay, went hand in hand with microscopy. Everybody was trying to build better, more powerful, more resolved lenses, okay, so they could view that microscopic world to a much, 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 much greater degree. So it went hand in hand. Microscopy and the discovery and early on investigation of cells, the history of it kind of went hand in hand. Over time, scientists were able to construct better and better and better microscopes. The microscopes that we're going to use in this unit and that are commonly used today are compound light microscopes, where we're using two lenses to actually magnify an object. Magnification is a vocabulary term that we need to know that refers to the quality of making an image appear larger than its actual size. Okay, so magnification, making an image or making an object appear larger than its actual size. Resolution, we're talking clarity. Okay, so we can make something look really, 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 really big, but it can be really fuzzy. The view can be obstructed. It's not very clear. Resolution is the clarity of that image. So where magnification, we're talking about making it bigger. When we're talking about increasing the resolution or the clarity, we're talking about making the picture clearer. Okay? Compound light microscopes do both. They use a light source to shine it through the actual specimen or sample on the microscope slide, whether it's prepared or whether you are preparing it yourself. And then that image is magnified by one of the initial objective lenses and then transferred through the body tube up to the ocular lens or the eyepiece and then further magnified and made available to you to see. Okay? We'll talk general microscope structure and function in class here early on in this unit. In addition to the compound light microscope, we've had several other technological advancements that have allowed us to view the microscopic world even further. Okay? We're talking electron microscopes, electron light microscopes, where we actually take a beam of electrons and shoot them at the specimen or the sample on the slide. Okay? Now, we're not going to get into a lot of the nuts and bolts on how an electron light microscope works or what's taking place, but there are two major types. Okay? An electron scanning microscope and an electron transmission microscope. All right? The transmission microscope is going to take the electrons and it's actually going to shoot it directly through the material, giving you a 2D image. Whereas the scanning is going to go ahead and it's going to take the electrons, it's going to focus them on the specimen, and they're going to bounce off. And through the software and the technology that's involved, it actually can create a three-dimensional image of that object. So electron light microscopes give you magnification that compound light microscopes cannot come close to, and there's two types. There's the electron transmission microscope that gives you a two-dimensional view of the object, and an electron scanning microscope that gives you a three-dimensional view of the object. Okay, so we've got topic one out of the way. We've talked a little bit about the discovery of cells, all right, or the coining of the term with Robert Hooke. We talked about the further investigation and discovery with Anton von Leeuwenhoek and discovery of animalcules. And we've also talked about how the microscope has changed over time and gotten to what it has gotten to today, the compound light microscope and your electron light microscopes. Now we're going to go ahead and we're going to talk about the cell theory. So this early discovery and investigation from the likes of Hooke, von Leeuwenhoek, and all of their other comrades, and how it led into further investigations by German botanist Matthias Schleiden, German zoologist Theodor Schwann, and German physician Rudolf Virchow. Okay? So three individuals are heavily credited with the initial finding of the cell theory. Okay? We stated earlier in the video that the cell is the smallest structural and functional unit of life. It's self-sustaining, the smallest structural and functional self-sustaining unit of life. These gentlemen, with their findings, using microscopes in each other and bouncing off ideas, came up with 3 foundational principles to help us declare that the cell is indeed the smallest structural and functional self-sustaining unit of life. The first part of the cell theory is that all living things are made up of one or more cells. All living things made up of one or more living cells. Part 2, cells are the basic units of structure and function in organisms. You've heard me say that already a couple times here. Cells are the basic units of structure and function in organisms. And part 3, all cells come from pre-existing cells. That, ladies and gentlemen, is the cell theory, and it helps form the foundation for a lot of other biological investigations and sub-studies that have been further advanced over the last 150 years. The cell theory itself pretty much developed anywhere between 1830 to 1860. Schleiden, Schwann, Virchow, really credited with it, but there were a slew of other individuals that were assistants and helped contribute to the actual discovery and nailing down of the cell theory. It's a little more broad now, but we are only going to go into those 3 pieces. All right? What were they again? The 3 pieces of the cell theory? Think for a second. All living things are composed of one or more cells. Cells are the basic units of structure and function, and all cells come from pre-existing cells. Okay? Alrighty. Video topic 2 in the bag now. The cell theory and a little more history on cellular discovery and cellular investigation. Now we're going to get into the 2 common types of cells. The 2 common, smallest structural and functional life-sustaining units. Self-sustaining units of life. Okay? Prokaryotes and eukaryotes. Prokaryotes have been around for 3.5 billion years. All right? These were the first organisms to actually live on planet Earth. They are single-celled living things. So a prokaryote is a single-celled organism, and is typically a little more simple than that of its eukaryotic brethren. Okay? A eukaryote, on the other hand, is a multicellular organism. Now, not all eukaryotes don't have to be multicelled, so I'm going to get a little confusing here in a second. Okay? Whether you're a proc or a prokaryote or a eukaryote, you share some common characteristics. Those common characteristics being a few of the C's: cytoskeleton, cytoplasm, cell membrane, okay, and then ribosomes. All right? You also share, what should I say here? I don't want to say DNA, because not all cells possess DNA, but I guess genetic information. All right? So 5 common features: cell membrane, cytoplasm, cytoskeleton, ribosomes, and genetic information. Okay? Whether you're a prokaryote or eukaryote, you've got those 5. Cytoplasm is kind of like the jelly-like watery substance that helps to suspend everything inside of the cell. It's not so much protection as it is just kind of like a sludge that, again, kind of helps keep things where they are. It's also a fluid material, so it assists in moving things from one place to the other. It gives items a medium at which to move through. So it kind of provides a little resistance to almost aid in movement of materials throughout the cell. Cytoplasm. Root word cyto meaning cell, plasm meaning water, a fluid. The cytoskeleton is the cell skeleton. Cyto, cell, skeleton, a skeleton. You've got a various network of protein fibers that are running themselves through the cell, helping provide a rigorous structure for the cell so it just doesn't collapse in on itself, and also assisting with transport of some materials and some organelles or some other entities in and around and throughout the cell, the cytoskeleton. Okay? The cell membrane is kind of like our bouncer. It surrounds the cell all right, and separates everything that's inside of the cell from everything that's outside of the cell. It's a phospholipid bilayer. We're going to look at it in much greater detail next class period. But the cell membrane is a common feature between prokes and euks. And it helps keep things out that need to stay out, and it helps keep things in that need to stay in, and it controls or regulates what enters and what exits. Ribosomes, we've seen these, are specialized proteins, okay, that assist in making other proteins. So they have a little bit of genetic information that's attached to them, a set of instructions, if you will, and it's kind of a protein that helps make other proteins. That's what ribosomes are. Again, proteins are the structural and functional backbone, okay, to everything at the cellular level. We studied those with macromolecules. And ribosomes assist in making more of those proteins that can then make more proteins that can then make more proteins and keep on helping the cell run and do what it needs to do, structurally and functionally. Finally, genetic information. All cells, whether you're a euc or a proc, have some type of nucleic acid present. It could be DNA, it could be RNA, doesn't really matter. If you're a proc or a euc, you've got genetic info. You have some instructions that tell your cell what to do at that level to sustain life. Those are the 5 common features that are present in both prokaryotes and eukaryotes. So those are the similarities. Those are the commonalities. Now the major differences. We've already stated one, prokaryotes are single-celled organisms. Great examples being bacteria. So if you're a bacterium, you're a prokaryote. All right? You're a single-celled organism, and you may have some additional features to assist in sustaining life. You could have what we call a capsule, which is typically a fat and sugar-based encasement around the cell to help aid in structure and protection. You could have a cell wall, all right? A cellulose concentrated material that surrounds the cell membrane to again aid in structure and protection. And you could even have maybe some hair-like structures surrounding the outside of you, cilia, or a corkscrew tail-like structure known as a flagellum. All right, again, 2 things that could assist in a single-celled organism like a bacterium to move itself through some type of fluid aqueous environment. So a little bit about prokaryotes there. Been around a really, really long time and is still alive and kicking today here on planet Earth. Eukaryotes, on the other hand, the major difference that they have is that they have a membrane-bound organelle. Okay? Prokaryotes do not have membrane-bound organelles, meaning they do not have separate internal compartments within them that have cell membranes surrounding them. Eukaryotes do. They've got items like nuclei. They have items like mitochondria, Golgi bodies, endoplasmic reticula. They've also got lysosomes, vesicles, chloroplasts. Okay? Eukaryotes have organelles. They have separate little internal compartments that have cell membranes surrounding them that help to regulate what enters and exits that little material, and then has that little compartment carry out a specific function or specific job. So the major difference between procs and eucs, eukaryotes have organelles, membrane-bound internalized compartments. Prokaryotes do not. Okay? Typical eukaryotes would be animal cells, plant cells. Okay? You could also have some single-celled eukaryotic organisms. All right? Stuff like protists. But most of the time when you're looking at a eukaryote, you're talking an animal or a plant. And most of the time you're talking about an organism that is multi-celled, has many cells, not just one. Okay, so that does it here for our Lesson 1 video, a little introduction to the cells, a little cellular information at a glance. Know that we've talked about the history, okay, the early discovery and early investigation into cells regarding Robert Hooke and the coining of the term, Anton von Leeuwenhoek, the father of microscopy, and how he first started to see some of these little guys. And we also talked about how that history parlayed into the development of the cell theory, our second major concept. Those 3 little foundational principles, concepts that gave way to many other sub-studies in the field of biology. We looked a little bit at the compound light microscope and electron light microscopes, the 2 microscopes, the 2 instruments we use to analyze the cellular world and to continue to learn about it and investigate it. And then last but not least, our third video topic was eukaryotes and prokaryotes, the two common types of cells. How are they similar and how are they different? If you have any questions, by all means, shoot me an email, tweet me, contact me in some way or another. Just make sure you got a little bit of this information down. I'll see you guys in class.